welcome back to the new video. And in this video we are going to talk about the Kingdom Monera. Okay, so the sole member of Kingdom Monera are the bacteria. Okay, so the Kingdom Monera contains bacteria. Okay, so now let's talk about the general characteristics of the species belonging to Kingdom Monera. Okay, so let's here write about the general characteristics. Okay, so the Kingdom Monera, they are prokaryotic. Okay, so what does it means? That is, they lack the well-defined nucleus. Okay, so lack well-defined nucleus. So this is the typical feature of a prokaryotic organism. Okay, and DNA do exist there in the prokaryotes, but it exists as a nucleoid. Okay, so DNA exists in the form of nucleoid. Okay, and the third feature of the prokaryotes are it lack membrane bound organelles. Okay, so lack of membrane bound organelles. Okay, so what organelles they are lacking? Like mitochondria, endoplasmic reticulum, Golgi apparatus, etc. Okay, so the first feature of the species, that is the bacteria that are included in Kingdom Monera, is include the prokaryotic feature, that they lack the well-defined nucleus. DNA exists in the form of nucleoid, and they lack the membrane bound organelles. Okay, clear? Now the bacteria, they are unicellular. Okay, this is the second feature. The third feature is that the cell wall is present. Okay, however, the composition of the cell wall, okay, varies. For example, the eubacteria, it is made up of peptidoglycan. Okay, so eubacteria, they have cell wall that is composed of the peptidoglycan, whereas the Archaebacteria possess the cell wall that is made up of polysaccharide and proteins. Okay, so the bacteria do possess the cell wall, but it differ in the composition. Okay, and mycoplasma, they lack the cell wall. Okay, if we talk about the mode of nutrition, then the Kingdom Monera exhibit highly diversional nutritional mode. Okay, they can be autotrophic, that is, they can synthesize their own food, or they can be heterotrophic. Okay, or they will consume the food made by others. So if they are autotrophic, they can be photosynthetic, either they can make their own food through the process of the photosynthesis, or they can be chemosynthetic. Okay, okay, the chemosynthetic mode of nutrition involve oxidizing the inorganic substances, okay, to obtain the energy. And if they are heterotrophic, they can be saprophytic or parasitic. Okay, okay, so this is all about the nutrition. Now let's talk about how the Kingdom Monera species, they reproduce. So the reproduction is basically asexual. Okay, so they divide through the asexual mode of reproduction, and the most common one is the binary fission. Okay, budding. Okay, and there are some bacteria that undergo a form of genetic recombination through conjugation, okay, transformation or the transduction. Now if we talk about the size of the species belonging to the Kingdom Monera, then they are microscopic. Okay, the size is microscopic. We have to use the microscope to view them. They are so tiny. Okay, and the size ranges from 0.2 micrometer to 10 micrometer. Okay, this is the size range. Now I already told you about the eubacteria and archaebacteria. So basically there are major group, okay, within Monera. So the two major groups are Archaebacteria and eubacteria. Okay, so the Archaebacteria, they are the ancient bacteria. Okay, and these Archaebacteria, they are very special. Okay, so why they are special? Because they can live in extreme environment. Okay, they can survive in extreme environment. And what kind of extreme environment? They can be survive the extremes, like having high concentration of salts. Okay, so they can survive in the salty area, they can survive in the hot springs, and they can survive in the marshy area also. And how they can survive? Because they have the cell wall. Okay, so cell wall will protect them. Okay, so as I told you here, the cell wall of the Archaebacteria, it lacks peptidoglycan and is composed of the polysaccharide and the protein. Okay, so basically there are three types of Archaebacteria. Okay, the first one is methanogens. Okay, so these methanogens, they are found in the marshy area. Okay, and in the guts of the ruminant animals like cows and buffalo, and they produce methane. Okay, the biogas from the dungs. Okay, the second type of Archaebacteria is thermoacidophiles. So from the name it is clear that these bacteria, they will grow in the high temperature and acidic condition. Okay, so they thrives in the hot acidic environment, example hot spring or the sulfur springs. Then comes the third types of Archaebacteria, that is the halophytes. Halophiles, not phyte means plants, so they are halophiles. Philes means loving, so halo means salty condition. So the bacteria that are growing in the salty conditions, okay, extreme salty condition like salt lakes, they are known as halophiles. Now let's talk about the eubacteria. So there are thousands of eubacteria, okay, and they are characterized by the cell wall containing the peptidoglycan. Okay, and these eubacteria, they can be motile and they are also non-motile. Okay, it means that they can move or not. They are like they cannot move also, motile, and they exhibit non-motile also, and they are of diverse shape. Okay, so diverse shape. So let's talk about the shape. So basically there are four kinds. The one is the cocci. So cocci is the spherical shape. Okay, so these kind of bacteria, they are like this sphere kind of. There are other second kind of bacteria, that is bacilli. Bacilli means they are rod shaped. Okay, So the rod shaped bacteria, they will look like the rods. Okay. Then there are third kind is the vibrio. They look like a comma. Okay. So they are comma shaped. So they will look like a comma. And then there is a spirella, the fourth form. So the name itself clear that they look like a spiral. Okay, so they will look like this. Okay, so there is a great diversity in their shape. Okay, how they look. So example is E. coli, the example of eubacteria. The eubacteria are also known as the true bacteria. Okay, so if somebody asks you that which category of bacteria are true bacteria, then ancient bacteria are the Archaebacteria and eubacteria is the true bacteria. Okay, so some example of the eubacteria involves E. coli. Okay, this is present in the human gut. Okay, then there is another example is the Streptococcus pneumoniae. Okay, this causes the pneumonia. Streptococcus, coccus means they are round in the spherical in the shape. Okay, and they cause the pneumonia in human being. Okay. Okay, then there are cyanobacteria, and these cyanobacteria, the important thing about them is they are photosynthetic. Okay, autotrophs, it means they will prepare their own food. Okay, so for the photosynthesis we need chlorophyll, right? So they possess the chlorophyll also. Chlorophyll A they have. Then they can be cyanobacteria, they can be unicellular. Okay, colonial. Okay, they can be in the colony or filamental. Okay, the next interesting thing about the cyanobacteria also is that they can fix, some of them can fix atmospheric nitrogen also. Okay, so they can, some of them, okay, they can fix atmospheric nitrogen. Okay, and to fix this they have the special kind of cells, they are known as heterocyst. Okay, so the example is the nostoc and anabina. Okay, so cyanobacteria often have the mucilaginous sheath also. Now let's talk about the mycoplasma. So these mycoplasma, they lack the cell wall. Okay, and these are the smallest known living cells. Okay, so if somebody asks you that which is the smallest known living cell, then you have to write the mycoplasma. Okay, and these mycoplasma, they can be pathogenic also. It means they can cause the disease in human beings. Okay, and they are pleomorphic. Pleomorphic means they will exhibit also the great pleomorphic, they will exhibit the great variation in their morphology, that is in their shape. Okay, now let's talk about the importance of eubacteria, why the eubacteria are beneficial or important for us. We will discuss here one by one. First of all, they are very good decomposers. Okay, so they play a crucial role in the nutrient cycle. Okay, and I told you earlier also that some of them, they help in the nitrogen fixation also. Okay, so they can convert the atmospheric nitrogen into usable form for the other plants, for example the rhizobium. Okay, then the third is the food production. So the several bacteria, they are used in the production of food like yogurt, cheese, fermented food. Okay, example lactobacillus. Okay, then the fourth is they have the industrial application also. Okay, What kind of industrial application? Certain eubacteria, they are used in the production of antibiotic, vitamins and enzyme. Example, streptomyces. It is used for production of the antibiotics. Okay. Then there are some pathogenic strains also. Okay. So these can cause some diseases in human being. For example, Salmonella enterica, it will cause the food poisoning. Okay. And there are certain bacteria that are help in the that will help us in the bioremediation. The bioremediation means they are used to clean up the pollutant. Okay, so they are pollutant cleaner. Okay, so now quickly let's talk about the structure of the eubacteria. So I will just write the name so that it is easier for you to remember. So the eubacteria, they will have the nucleate. Okay, I already told you that the DNA exists in the form of nucleoid. Okay, then there is the plasmid. So plasmid is the extra chromosomal DNA, okay, often carrying the gene for the antibiotic resistance. Then the eubacteria also possess the ribosomes, and ribosomes are the site of the protein synthesis, that is 70S. Then they have the cell wall, okay, for providing the rigidity, the structural support, okay, and they have the cell membrane, okay. So cell membrane, it will regulate the substance passages, okay. Then they have the flagella for motility in few species. I already told you some can be motile and some are non-motile. Okay, so those that are motile, they have the flagella. Then they have the pili, pili. So pili is a hair-like appendage for attachment or conjugation. Okay, and the last they have the capsule. Okay, so capsule is a slimy outer layer for the protection and the adherence. Okay. So this is all about the key structure of the bacteria, and before closing the this video, I will keep it very short. Let's talk about the reproduction mode. So just quickly we'll talk about the reproduction. So reproduction is, I already told you, it is basically through the binary fission. Okay, so this is the most common form. In this, the cell duplicates its DNA and then divide into two identical daughter cells. Okay, and there is conjugation also. So in the conjugation process, the transfer of genetic material occur between the two bacterial cells through a conjugation tube. Okay, then the transformation process, the transformation process involves the uptake of DNA from its surrounding, and then the fourth one is the transduction. Okay, so transduction is the transfer of bacterial DNA, okay, by a bacteriophage. So this is all about the Kingdom Monera you need to know, okay, in this video. Thank you for watching the video.